everyone, I'm AuBree LaForce.
And hello, I'm Jasmine Barker.
And we're here to talk to you today about something I think we're all pretty familiar with, especially around this time of year. So to start off, I have a question for you, and yes, you can actually answer. And it is, what do finals, money, and relationships all have in common? Stress, yes, exactly, stress. These three things cause stress, as we all know, along with other things in our lives. And how we respond to stress is we have an integrated stress response. And that is caused because when we undergo stressful conditions, we still want to be able to survive. Some things happen in our body that respond to the stress that are negative. However, we still have to survive under these stressful conditions, and that is where we have our integrated stress response. Now, what's important is our body undergoes this integrated stress response to protect ourselves and to still survive. However, there are very negative side effects of this, one being that it affects our memory consolidation. And what that means is it affects our ability to go short-term to long-term memory. And I know some of us want to be researchers and doctors and teachers, and we want to remember our mom and dad's birthdays. So, memory is really important in life. So, as we said before, the clicker, there we go. Okay. So as we said before, when you're under stressful conditions, your body undergoes the integrated stress response. And this occurs by one, you get sensor activation. So these are when sensors in your body realizes that you're under stress, and then they become activated. Then it leads to a phosphorylation cascade. Different phosphorylation events happen and trigger different kinases, and this affects different transcription factors. And then you have an upregulation of specific mRNAs and naked RNAs that allow your body to cope with this stress. So all of these things overall lead to a decrease in normal protein synthesis in your body. And as we all know, protein synthesis, we need proteins to do anything in life. And therefore, this decrease in protein synthesis is a very negative cause of this integrated stress response. However, what's important to note is when this protein synthesis is stopping, you also have an upregulation of your specific mRNAs at the same time. So although protein synthesis is decreasing, your body responding to the stress upregulates the specific mRNAs in order for your body to survive. So the question is, what can stop the negative effects of this integrated stress response? And this is what Sadowski and his colleagues were out to test. They wanted to see what they can do and what they can find that will inhibit the negative effects of this stress response, and therefore leading to that memory consolidation and leading to your ability to still transition between your short-term and your long-term memory. And that's when they found the ISRIB. And for the rest of the presentation, we're going to call this RIB.
Okay, but before getting started and looking more into their studies, I'm going to give you a little bit better of an understanding of what exactly your body does to respond to stress. So when you're stressed, you start to accumulate a lot of misfolded proteins in your endoplasmic reticulum, which are in here in red. So once they start to accumulate, you have sensors that will recognize these and become activated, which are the IRE1, the ATF6, and PERK. But for the majority of our presentation, we're going to focus on PERK and their downstream effects.
So going past that, why does stress cause misfolded proteins? What's sort of the step right before those misfolded proteins? What do you think? Somebody from the audience.
Release of something like a hormone or a molecule?
It will be a molecule, okay? So what do you think? What would potentially cause a protein to misfold?
Something binds to it, or something like else binds to misshape it.
So you get some other interactions that aren't as responsive. So then, what could that be?
A fight or flight response, like sort of like all those hormones being released. I don't know.
So what's that gonna cause? So the hormones themselves are basically signaling. They're gonna make proteins that wouldn't really cause proteins to misfold.
Maybe phosphorylation.
Okay, you're getting closer. It's not necessarily phosphorylation, but think about, the closest thing that I can think about Alzheimer's. So in Alzheimer's, what's the big thing that causes misfolded proteins? Free radicals. Free radicals are formed when you're stressed. Okay, you actually have a lot of free radicals forming when you are stressed. Those can react with proteins, causing them to misfold, and now you have a bunch of these misfolded proteins that aren't doing you change the structure, you no longer have that function. And so that's what's leading to the accumulation of these misfolded proteins.
So once these misfolded proteins accumulate, your PERK sensor will be phosphorylated or activated. And from here, this will cause an autophosphorylation of your ELF2 alpha. So once ELF2 alpha is phosphorylated, it actually activates the ATF4, or increases the production of ATF4. So ATF4 is a transcription factor and is one of the largest and most important transcription factors. So it regulates the whole transcription network. So once ATF4 is increased, it'll upregulate the CHOP or the GAD. And this is what causes the downregulation of the protein synthesis or the upregulation of the mRNA. So
Now, what's important to note here is that when you have your upregulation of your mRNAs and your naked mRNAs, those RNA binding proteins, they accumulate into stress granules.
And stress granules are actually positive for the cells when they're trying to survive under stressful conditions because they protect the mRNA from chemicals or just harmful situations. But as looking into memory consolidation, it's negative because this accumulation of the mRNA prevents you from developing long-term memory.
And we'll go into this further in detail a little bit later, but just to give you an idea, so the stress granules, when you need to go from short-term to long-term memory, it's when your neurons have to fire and are stronger firing of your neurons between synapses. And then those firing of the neurons, they happen in between channels. And your actual channels are formed from your proteins. So therefore, if you have a decrease in protein synthesis and these stress granules form, therefore you can't form proper channels, and that's when you're unable to make those strong firings between and that transition between short-term and long-term memory, the stronger firings in the neuron. So that's how it all is kind of connected. So as we said before, you have your integrative stress response caused by your stress, overall decreasing protein synthesis. Now, Arsadrowski and his colleagues, they wanted to see what can keep this protein synthesis going, but while stopping your integrative stress response, or seeing which part of the integrative stress response is the most important to stop. So first, during their test, they wanted to test, in order to complete your test, they had to have a stable cell line. And now in order to make a stable cell line, they wanted to produce cells that express this ATF4. So as we showed before, that last step in that phosphorylation cascade was the production of ATF4. Therefore, they want to make a stable cell line that showed the ATF4, that had the ATF4 in the DNA. So then they changed the DNA to have this ATF4. And the first study that they did to get a stable cell line is that they tagged ATF4 with luciferase. However, luciferase doesn't fluoresce on its own, and so it only fluoresces with presence of luciferin. So when they gave the cells luciferin and then they ran their fluorescence test, everything turned green. So it was really hard for them to see where the actual cells were and which cells actually had the ATF4. So therefore, their next test, they presented the cells with ATF4 tagged with GFP. And then since GFP can fluoresce on its own, when they did the fluorescence test, just the cells turned green, and they were able to establish this stable cell line, which is their HEK293T. And that's a cell line that they used throughout the rest of the experiment.
So once they were able to detect and make a successful cell line, they used these cells to test various compounds and run a screening to see which compounds were best at preventing ATF4 production. So their idea with this is the fact that if you were to activate the PERK sensor, you would also cause the production of ATF4. So if you're able to inhibit ATF4 production, it means that you're preventing the IRS response or at the time they thought preventing the PERK sensor from being activated. So after running various compounds, they decided that what we refer to as RIB to be the best compound, but they realized that RIB is an isomer, so they wanted to see whether or not the cis or trans isomer is what causes this inhibition or prevention of the ATF4 production. So they ran the same kind of
Let me go
test.
back one. So, if you're an organic chemist, this is a really neat molecule. Why is this a really neat molecule? Focusing more on the trans than the cis.
This looks cool.
It does look cool.
It's symmetric.
It's very symmetric. I mean, extremely symmetric. If you look at that, that is completely exactly the same. You flip it around. So very, very symmetric. And what does that suggest about the PERK binding site then?
It's symmetric.
So it has to be symmetric, or you have multiple binding sites that are very close together that you're able to bind and inhibit both at the same time. And so because it's symmetric and it's able to bind and it decreases the activity of the PERK, it's suggesting there might be multiple subunits that you're able to inhibit at the same time, and they're very close together. So does that make sense? That makes sense. So, most likely what you're going to, and they actually showed this in experimental biology that the binding site for this was right at the cleft where you had two subunits coming together, which is really, really, which
Also,
is,
it's like a quaternary protein.
yes. So it's more of a quaternary structure for this protein.
So after running similar tests, they realized that the trans isomer was a lot more effective in inhibiting ATF4 production. So they measured the IC50, which we learned in this class. So it's the ability the concentration that's required to inhibit 50% of the cells. And they realized that the trans was almost 100-fold more potent. So they used this trans isomer for the rest of the test.
So now that they know that this RIB, it inhibits your production of your ATF4. Now the next question that they proposed was, now that they know the inhibition of ATF4, they weren't really sure at which point the RIB was affecting. So was it affecting the phosphorylation of PERK up here, and is that what caused your decrease in your ATF4 production, or was it affecting your ALF2 alpha phosphorylation down here? So those are the two tests that they started out with because they wanted to see at which point was this ATF4 production being inhibited. So the first test they ran was an immunoblot assay, and they had where an antibody recognized the phosphorylation of PERK. And they ran this, and throughout the rest of their tests, they presented the cells with different stressors, so different ER stressors. And that is represented by your TM and your TG. Now what your TM does is it affects the end glycosylation, I believe is how you say it, glycosylation.
Glycosylation.
Glycosylation, great. And that is when, it makes the cells arrest in the G1 phase of the cell cycle. And then your TG, that depletes the cell of calcium and it inhibits your ATP, or your calcium ATPase. So those were two different ER stressors that they presented to the cells in order for them to be under stress throughout all of their experiments. So you'll see these TM and TGs. So under their immunoblot assay, you can see here, so this is our RIB, and then they wanted to see if PERK phosphorylation was inhibited. As you can see here, right here, the PERK phosphorylation was not inhibited by RIB. However, your ATF4 production was still decreased and still inhibited. So therefore, the PERK phosphorylation was not inhibited in order to decrease your ATF4. And secondly, as I said before, they wanted to see if the phosphorylation of your ALF2 alpha is what's causing this. And they saw that it was not the phosphorylation of your ALF2 alpha that inhibited your ATF4 production. As seen here, you have your stressors with your RIB, and you can see that there is no significant change with your RIB with the phosphorylation of your ALF2 alpha.
And what were the XBP ones that were on the previous So you talked about the PERK and the ATF, what were the XBP ones?
That was, I believe that was the specific, they mentioned it and they said that was, I can't remember off
So
the top
it-
of my head, but it was,
When ATF4,
oh, was it, is ATF4 cleaned at one point? No? I'm not sure.
so, anybody else? What's
I think
XBP
you
ones?
mentioned it.
What are the XBP ones?
I think you mentioned it, I think, in the, at the bottom of the figure, document, the article.
Yeah.
Right under the, what? Is it one of the kinases? No.
It's not just the control, is it?
Not in this case.
I believe it says it under the.
So what are the XBP ones?
Does it say that?
I'm not saying anything in the video.
Is it just a binding protein?
cDNA? Is that what it is?
You're close. So these are going to basically be what's going to be activated when you are going through a stress
Stress,
response.
okay.
So these are activated.
Because
and-
they're in both.
Right, right. And so you're going to still see these being produced. So this is not what is inhibiting sort of this whole process of stress response.
So is that showing kind of, is it acting kind of as a control, just showing that this is gonna be produced when you're under these two stress conditions? Or not a control, but to show that these stress conditions are causing a response?
So this test is mainly to figure out, so we know that it inhibits PERK, so it's inhibiting sort of that process, but they want to understand, out of inhibiting PERK, what sort of downstream events are affected, and how is the whole stress response affected. And so initially you say, okay, PERK, the phosphorylation of it, isn't affected. We know that you can't make ATF4 now, okay? The XBP1s, those are actually the proteins that are your regulators that you're going to be making. And you still see you're able to make those regulating proteins. So it all is relating to this ATF4 that you're focusing on at this point. So that's really what this is laying out. Okay, so phosphorylation, no. Is it these regulators that you would then be making more of because you had all this misfolded protein? No. Is it ATF4 production? Yes.
And then we previously went over that slide and showed that it's not the L2 alpha phosphorylation either that inhibits the ATF4 production.
So next they wanted to see, since they realized alpha-2 alpha is still being phosphorylated and so is the PERK sensor, they wanted to look and see if there are more downstream effects that RIP's affecting. So they looked at the global translation attenuation. So normally if you're not under stressful conditions, which are illustrated in the first half of the graph, you'd have normal protein synthesis. So methionine was tagged and was used to monitor how much protein is being synthesized, since methionine is always present at the beginning. And when you're under stressful conditions, it shows that less methionine is present, so there's less protein synthesis, less proteins present. But whenever you add the stress, add RIB to your stressful situation, your methionine levels go back up. So it's showing that this RIB is actually able to allow for regular protein synthesis to occur and for to have the same response as if you weren't in a stressful situation.
And they already said this, but I want to see for the audience, why is methionine the best amino acid to use for these type of tests?
It starts the chain.
So if you think about any DNA, how do you know to start that sequence? It's AUG, which encodes for methionine. So no matter what the protein content is, you're always going to have methionine. So it's a good measure. Instead of saying, okay, well we're going to use, say, tryptophan. Well, tryptophan content can vary depending on the protein. Here, you're just looking at methionine. And methionine is usually a rare one that you're not going to see throughout a whole structure, but you will see it at the beginning. So it covers all the proteins being made. Good.
So overall, to summarize just what we went over so far, they realized that the RIB actually isn't inhibiting the IRS, but rather it's just preventing the cells from responding to the IRS, which is pretty interesting. So from there, since they realized that L2 alpha is still being phosphorylated and this, the rib isn't affected or doesn't respond to this, they wanted to see if you change the kinase that's phosphorylating the L2 alpha, if this has an effect on the ATF4 production. So when looking at the HRI, they realized that the PERK is still being phosphorylated as expected, but ATF4 production is decreasing. So this kinase had the same effect as the other one previously studied. And then they looked at amino acid starvation, which is another way to induce ER stress, and they realized that the PERK, once again, still being phosphorylated, but ATF4 production is decreasing. So no matter which kinase you use to phosphorylate L2 alpha, the rib is able to still prevent ATF4 production.
Okay, so then, as we said before, your ATF4, it is a transcription factor, and it regulates a whole transcription network. And so, as said at the beginning, when we went through the phosphorylation cascade all the way down to the bottom, the last step was when this ATF4, it works together with CHOP and our GAD34, and it activates these two transcription factors. And these two are transcription factors that allow us to respond and still survive under stressful conditions, because these are the mRNAs. So the next test they wanted to see is that does our rib inhibit the production of our CHOP and our GAD? Their prediction was that it was going to, because these two are controlled by our ATF4 production. And as they found, it definitely did inhibit the production of these two through the inhibition of your ATF4. And as seen here, it's just like they're showing the fold change, and this is your CHOP and this is your GAD with presence of your rib in these two columns.
And in this case, the control is when you're already under stressful conditions. So you're under stressful conditions and then you add the rib to it. And this is more just of a visual, which is
Where's the
kind
visual?
of, yeah. Which one? I can-
Is it the top?
The bottom.
Okay, so this is more just of a visual aid. So whenever they luminesce CHOP, they use actin because DAPI would be in the nucleus.
No, they use DAPI in the next test
Yeah.
we're going to talk about, but they just used actin to identify where the cells were located.
So in the control, CHOP isn't present because you're under stressful conditions. Then when you add TG, you're under stressful conditions. CHOP's being produced because ATF4 is being produced through the integrated stress response. But then whenever you were to add rib, you actually prevented the production of CHOP, which was shown in the data previously.
You can leave the lights on.
Okay.
And then, so then the next thing that they wanted to test is whether or not this rib will affect the other sensors. So in the first slide we showed with the phosphorylation events, we had the three different sensors. You have your IRE, you have your ATF6, and you have your PERK. Now, up to this point, we've been talking a lot about PERK, but now that they wanted, since they already looked at the whole phosphorylation cascade that is caused by PERK, now they wanted to see, does this rib have any other effects on the other two sensors that they haven't looked at yet? So what they did is they studied the effect of rib on IRE1, which is another sensor. And so this is your stressful condition, and this is your stressful condition with your rib. And you can see, so what they're measuring is the tiny little dots. And this is, you can see that there is no decrease, and therefore the rib did not have an effect on this sensor. And then the other sensor they wanted to test was their ATF6 sensor, and if the phosphorylation and activation of this sensor was going to be inhibited or not by rib. And as shown here, you still have your ATF6 present with your stress in your rib, showing that the rib did not affect this ATF6. So up to this point, the rib has only affected the PERK sensor, not the ATF6 or the IRE1.
So previously, different studies have shown that when you inhibit the phosphorylation of the L2 alpha, this reduces the viability of cells. So to kind of see if the rib also has the same effect, they wanted to test to see if when adding rib, if you increase the risk of apoptosis. So as you can see from the control, there is minimal apoptosis, and they studied the caspase 3.7 because this is a protease, so the higher levels of the caspase, the higher risk of apoptosis, or the more apoptosis is occurring. So whenever you add stressful situations, the level of caspase increases as expected because you're under stressful conditions, your cells may undergo apoptosis. When adding the stressful situation and rib, though, the apoptosis or the level of caspase increased dramatically. And they believe this could be due to the fact that if your body is not responding to your integrated stress response, it's not going to eventually go back to its regulated state. So if it continues to just not respond, eventually it may undergo apoptosis. So this is one of the side effects.
And to kind of visualize this, so your rib is said to decrease your ATF4, and it's said to allow protein synthesis to keep occurring. So as you keep synthesizing proteins, more and more proteins are not responding to that integrated stress response in presence of your rib, that's when you undergo apoptosis.
Is that all clear for everybody? Sort of that? Because, I mean, if you look at that initially, it doesn't make any sense. You wouldn't expect your cells to be more apoptotic in the presence of this inhibitor. How about somebody from the audience go through that for us?
Maybe if like you make more of the protein than your cells, like wondering if it might like over accumulate, so then would that trigger some apoptosis?
So think about what's the very first step when you have the integrated stress response activated. In your cell, the very first thing that it does is shuts down protein production. Because you have all these misfolded proteins, you don't want to waste energy basically causing more and more proteins to be produced when they're all just going to misfold. You know, it's no sense wasting all that material. So you downregulate that, you upregulate all of these sort of regulators to help you fold and basically get the system under control. Now rib, in this case, is inhibiting the ability of you to induce that stress response. So is that going to cause you to have more or less misfolded proteins in the presence of rib? More, if you're stressed and you have rib, so that's that TG plus the rib, sort of the upside down triangle is that blue. So here you had a stress, you're not able to stop production of those misfolded proteins because you have rib present. So you're going to have more and more of these misfolded proteins. At that point, you're going to activate your caspase cascade to start to say, okay, this cell is there's something wrong with this cell. It needs to go through programmed cell death. Okay, that all makes sense? That's what they described, and I want to make sure everybody that was pretty clear because it's not a trivial thing to get, but good.
Okay, now just to recap where we're at, we've talked about a lot of phosphorylation events and a lot of things that caused other events. So right now, in our presentation and through their research, so you have your PERT sensor is activated when you're under stressful conditions, and this causes the phosphorylation of your alpha to alpha. Now this increases your ATF4 production. Now once that ATF4 production increases, it is the head of a transcriptional network, and it upregulates your transcription factors, which we talked about, your CHOP and your GAP, and it downregulates your protein synthesis. That leaves you with your naked mRNAs and RNA binding proteins, which eventually leads to your formulation of your stress granules. Now, the stress granules, as we said before, is kind of what leads to your inability to go from your short-term to your long-term memory. So therefore, their next question is they wanted to see what can prevent these stress granules and how these stress granules cannot be formed by inhibiting and looking at the effects of your alpha-2A phosphorylation.
Okay, so they looked at two different ways in which stress granules can be formed to see if it's alpha-2-alpha phosphorylation dependent or independent. So first, if you prevent the phosphorylation of alpha-2-alpha, as expected, stress granules would not form. There wouldn't be an accumulation of mRNA. But also, if you inhibit L4-alpha, which is actually used to go into the stress granules, so it's one of the mRNA present, you would never produce stress granules as well. So they look to see if you look at the pad A or the hip, if ribs able to minimize stress granules formed through those, or if it's only able to inhibit stress granules formed through ELF2 alpha phosphorylation.
And so then they looked at the formation of your stress granules, and in order to visualize, they used ELF3A or ELF3 alpha and our DAPI. So our DAPI is just used to visualize the cells, and our ELF3A or ELF3 alpha is present in stress granules. And so, as shown here, it might be, okay, so we had a little bit of a hard time visualizing this, so it might be hard to see, especially up here on the screen. But what they tested, the stress granules are these the little green. It's not like the big overall green. It's these tiny green dots are the stress granules. And so you have your stressful conditions, and then you have in presence of your rib. And so this shows that there is a decrease in the tiny green dots with the presence of rib under your ELF2 alpha phosphorylation. And then this is just the quantitative data for that, showing that your presence of rib has a significant decrease in your stress granule formation under the ELF2 alpha phosphorylation, what that phosphorylation causes.
And so what was ours? ARS?
That is a stress condition that is caused by your ELF2 alpha phosphorylation. So they're testing to see if the stress granule formation is dependent on your ELF2 alpha phosphorylation. Yes?
Why didn't they use the TM or the TG like they were before? Why did they start using one?
Because the TM and TG, I believe, are just like overall ER stressors. They're not specific to the ELF2 alpha phosphorylation, because they're looking specifically, does stress granules form from your ELF2 alpha phosphorylation compared to your ELF4 alpha inhibition. So I think they want to like specifically see. Does that make sense? Because the TM or the TG.
Well, they're using the TG.
I mean the TN, I don't think is as specific as the ARS. Is that?
Yeah.
Changed that. It wasn't.
And I know TG is a stronger inducer, the stress inducer, so maybe that may have something to do with it.
Yeah, TG
TG
is
is a lot
strong.
stronger, so I don't know if TM is strong enough to give the stress granule response they're looking for.
It wasn't the same.
Yeah.
Could you turn on the lights, please?
Yeah.
It was the same set of.
To the top.
Okay. I'll get it, guys.
Yeah, you want the TG because you want to be able to see the granules being able to be formed. And so TG, as they said, is a very strong inducer, so you can see the induction. TM is not a strong enough inducer of ER stress to form granules. It'll still shut down sort of protein production, but it won't form the granules. Now the R's, that I have to look up because I'm not. Anybody else know what R's does?
It's a use inducer of ELF2 alpha phosphorylation.
Okay,
So that's it.
so it's another strong inducer.
Okay, since they're testing like in this case is specific, and you'll see like the other one they used to test specifically the alpha and alpha inhibition. The next slide.
Okay. Any other questions? Does that make sense? Okay.
So then next, as we said, they wanted to test. So this is the quantitative for what we just looked at.
Is that the last fluorescent one that you have?
The next one. No,
Next
we have
one.
one more.
Okay,
Yes.
okay.
Okay, and so then the next test they did, we'll show you the graph first and then the fluorescence. The next one, they wanted to see the PAT A in your hip. Those are L4 alpha inhibitors, and so they wanted to see if RIB had an effect on those, those phosphorylations, and if those phosphorylations cause stress granules. So as shown here, so they used just how they use ARS. So the R's was a inducer for L2 alpha phosphorylation. Now your PAT A is an inducer for your L4 alpha inhibition. So those are the two things that they're comparing here. And shown here is you can still see the presence of those tiny green dots and those stress granules in the presence of RIB. Therefore, it was concluded that your RIB did not have an effect on your L4 alpha inhibition, and it only affected the stress granules that were formed from your L2 alpha. So the next thing we want to talk about is, why are we talking about stress granules? Why do we care? And what does this have to do with memory?
Okay, so memory consolidation is your ability to go from short-term memory to long-term memory. And to accomplish this, first you have to have increased synapse strength to be able to fire your signals, and also you need to be able to have your neurons repeatedly fire and be able to fire in like similar sequences. But whenever you're under stressful situations, you develop stress granules, so this accumulation of mRNA, your protein's not being synthesized. So as AuBree touched on earlier in the presentation, your actual channels that between each neuron, you require proteins. So if you don't have enough proteins, your signal's not being able to efficiently be fired from neuron to neuron. So this prevents long-term memory from being developed. So overall, big picture, this research is really important because under stressful conditions, we're unable to retain information, which is obviously really inconvenient, especially with finals coming up. So if they're able to figure out how to still allow long-term memory to occur although your cells are experiencing stress, it'd be very beneficial.
So now that they made these conclusions with these different tests, they wanted to see how it's going to affect memory in mice. So the heterozygote mice they presented, they said that they have enhanced memory. However, with the L2 alpha phosphorylation in presence of PKR, which is an L2 alpha kinase, that the brain cells, it impairs their memory. So they wanted to test this, and they actually did, and we hope you guys find it interesting too. They did a water maze along with some other tests. So to give you a little bit of a background, we have a video to show you of the Morris water maze that they used. Should I even try the volume? No? Okay.
So initially they have an elevated platform in the water maze, and they allow the mice to swim around and recognize this platform.
And there's only about an inch above the water that the platform sticks out. And so the mouse has to find the platform, get up on the platform, and realize that when it's on the platform, it'll be rescued from the water maze. So it has to swim around, find it, and then get up on it to be rescued. And then, as they showed, the water, after they trained the mice, then they put the mice back in, but they don't have the platform sticking out of the water, so the mice have to swim around and remember where the platform was and still be able to get on the platform.
And they also dye the water, so the mice aren't able to see where the platform would be.
So when they train the mice, they train them, they can see the platform even from under or over the water, and then when they test them, it makes it harder for them to see, and then it's not sticking out. So in their test that we're talking about, they use this water maze to test the memory of mice. And so what they did was they gave the mice this rib, and then they trained them through the water maze. And then they did this whole procedure, and they tested to see how long it took the mice that had rib and the mice that didn't have the presence of rib, and the different times it took for them to find the platform. Does everyone understand kind of how they did that? Okay.
We just found it really interesting, so we thought we showed it.
These are rats, right?
Yeah.
That's a big mouse, that's a.
Okay, so what they did, I said they injected the mice with this rib and they trained them through the water maze, and then they compared the effects of the mice treated with rib and mice that weren't. And they measured the latency or the time it took for the mice to recognize the platform. What's really interesting to note is that, so they did these over a period of, they trained the mice and then they tested over the 5 days. Now you can see that the mice that were normal and not treated with the rib, at first, the first 2 days, there's not too big of a difference in the time it took them to recognize the platform. Because, as we showed, that this affects long-term memory. It affects the ability for your short-term to be turned into long-term memories. So therefore, you can see that over right when they're trained, the short-term memory isn't, as nearly affected as the long-term memory is.
Okay, and then another test that was very interesting they completed was a tone shock. So they placed mice into a small area and they gave them a good high-pitched tone. And then right after the tone, they shocked the mice in the feet and they got them used to that stimulation. Well, then they took the ISR, IB, or the rib mice and compared them to just regular mice to see if when placed back into that same situation and they hear the tone, if they would freeze, if they would remember the shock that was associated with it. So, as this data shows, similar to the water maze, the rib are actually,
So this is
Yeah.
your short-term memory, and this is your long-term memory.
yeah, so they were a lot more effective in recognizing the tone shock and freezing for longer periods of time. And also in the short-term memory, it wasn't as much of a difference. So they were both able to remember the shock initially, but as time went on and you were to place the rib mice into the same situation, they were going to remember the shock and remember the pain associated with it.
So we definitely think, as we were looking at the different studies that they did with the mice, how important and how great of results they got with the mice studies, because this just really shows the effect that this rib can have on long-term memory. And so going forward with next research, Dr. Miller actually heard this guy talk at the biology conference, and he was talking about how his next step is to give the mice like blunt head trauma and to see if this rib can really reverse the effects and reverse the memory loss and to gain back memory. So it's really interesting, we thought, especially from the mouse studies, to see the different effects it has on memory. So overall, in conclusion, this rib is very beneficial going forward in studying what effects stress can have on your memory consolidation and making short-term memory go to long-term memory, and the importance it can have on having you remember even though you're stressed.
Any questions?
Cool.
Yeah.
So yeah, they are basically